Good day, everyone. On behalf of BioIT World's Global Web Symposia Series and our sponsor, Tecan, I'd like to welcome you to Advances in Non-Invasive Prenatal Screening with an NGS CE IVD workflow. My name is Elizabeth Lamb, and I'm the host and the technical director for today's event. I'd like to introduce our moderator for today, Nicholas Smith, Head of Global Marketing and Portfolio Management for Tecan. Welcome, Nick.
So good afternoon, good morning everybody, depending on your location. A very warm welcome from my side to this Tecan-sponsored web symposium. I think our topic today focuses on one of the most exciting areas of clinical diagnostics and how genomic technology is changing screening and diagnostic paradigms. Our partner today in this presentation is Premaitha Health plc. Premaitha is an international molecular diagnostics group whose primary focus is on non-invasive prenatal tests for pregnant women. And I'm delighted that the Chief Executive Officer of Premaitha, Dr. Stephen Little, is going to present for you today. Dr. Little holds a PhD from Heriot-Watt University in Edinburgh and has over 20 years' experience in senior positions in the diagnostic divisions of AstraZeneca and ICI. Prior to joining Premaitha, he was CEO of DXS, which was an innovative companion diagnostic company that was acquired by QIAGEN in 2009. So without further ado, I shall hand you over to Dr. Little. Thank you very much.
Right, thanks very much, Nick, and thanks to everyone for attending this seminar. I hope that you find it interesting. What I'm going to do this morning or this afternoon, depending on your vacation, I guess it's 4 o'clock in the afternoon in Manchester, is to talk a little bit about non-invasive prenatal testing. We'll spend a bit of time on how the technology works. So we'll start off with a kind of science section and go into some of the interesting aspects, at least I think the interesting aspects of the science. And then we'll talk about the clinical benefits and some of the limitations of the technology. And then at the end, we'll spend a few minutes thinking about where this technology might go next. And at that point, we want to stray well away from the science and move on to some clinical and indeed ethical issues. I'm just working on that at the moment. Oh yes, and I've remembered what I was taught by Elizabeth, who's our moderator, and she taught me how to do this yesterday. See, I've already forgotten. Apologises for that. Anyway, we're going to talk about non-invasive prenatal screening, and as the slide says, with a workflow that's been set up to IVD levels. And this first slide, Introduction to NIPT, really summarises the main points about the technology and why it's so important and why it's really taken off to be the most important application, at least the most important clinical diagnostic application of next generation sequencing. Prenatal screening for common trisomies, which is primarily Down syndrome, Edwards syndrome and Patau's syndrome, has been around for a long time. It's not new, the idea of getting a prenatal screening test. Even 10 years ago, there were millions of prenatal screening tests performed around the world, but there was a problem with them, and the problem with them was that they didn't work as well as they could do, causing false positives and false negatives. So over the last 3, 4, 5 years, there's been a major improvement in test performance through the introduction of what are called NIPT, which stands for non-invasive prenatal testing. And this technology analyzes DNA sequences from a sample of a pregnant mother's blood, and within that blood there's some DNA which has got the same genotype as the fetus. And put very simply, the benefit of NIPT tests is that they are more accurate than the existing technology, which is called the combined test. And when I say more accurate, I mean more specific, in that there are fewer false positives, and more sensitive, in which there are fewer false negatives. And when we think about a screening test, they are really the 2 key parameters that we want to maximize. And the only real disadvantage of NIPT, at least from a consumer perspective, is it's more expensive than existing prenatal tests. It's more expensive at the moment. It's not clear it'll be more expensive forever. My view and the view of many people around the world is it doesn't matter really. In the fullness of time, all prenatal screening will be done this way, though that is not the case at the moment. I'll tell you a little bit about our company, first of all, before we go into the science, the technology, and the clinical aspects. We're a molecular diagnostics group. We are interested in DNA analysis technology, and we could have, of course, developed assays for any test that we wished, but the one that got us very excited was non-invasive prenatal testing. We chose non-invasive prenatal testing. We thought it was a very good application, both clinically, because the clinical benefits I think are very clear, and we'll talk about those in a minute, and also commercially, because being a screening test, it's applicable to effectively all pregnancies throughout the world. The market size is absolutely enormous. And we launched in 2015, not the first NIPT test, but the first NIPT test that was available as an in vitro diagnostic product with a CE mark to demonstrate that. We're based in Manchester, but earlier this year we acquired another company called YourGene, and they're based in Taipei and Singapore. So we really have got pretty broad reach at the moment, and we have customers across the UK, Europe, Middle East, Southeast Asia, Russia and Africa. And they're our product customers, and we also have a service running out of Manchester and Taipei. The one place that we haven't really expanded our business at the moment is the USA. The FDA have set a high regulatory bar, and we haven't yet got round to jumping over that bar, but certainly the USA is part of our plans for the future. And to date we've performed over 80,000 non-invasive prenatal tests. In terms of our business model, our primary model is we sell a product, a kit, and the customers for our product are laboratories. So any laboratory would like to introduce non-invasive prenatal testing into their own lab and supply their own customers can buy the kit from us, some instruments from other companies, put the whole thing together along with control software and analysis software and customer-facing software, and we sell a complete system to allow a lab to offer that service itself. Additionally, in Taipei and in Manchester, we have the clinical service laboratory, so we are also able to receive samples into the lab and process them ourselves. And oftentimes, if it's early stages for a laboratory customer, they don't have the volume to justify the installation of the product, this is a way to get customers moving quickly. And then we have a third version of the test, and in this case we allow the customer to develop their own NGS method, which we then process remotely in Taipei and send the results back. So it's a kind of third way of doing the testing. That particular version of the test is not CE marked. So first of all, I'm going to talk a little bit about how NIPT works. I'm sure some of you know some of this already, but there's a couple of interesting points perhaps that will be that might be of interest to you. So the first thing is we often talk about fetal DNA in the maternal circulation. That's the first lie. It's not fetal DNA at all, really. The DNA that we find associated with the fetus in the maternal circulation comes from the placenta. Now, as the placenta and the fetus are derived from the same fertilized egg, it's exactly the same genotype most of the time, nearly all of the time. We're going to come back to that. But when people talk about circulating cell-free fetal DNA, they're really talking about circulating cell-free placental DNA. And around about, on average, 10% of the DNA in the circulation comes from the fetus, and about 90% comes from the mother. But that is quite variable, and it's an important variable in terms of test performance. Now, as you'll be aware, the cause of Down syndrome, which we use as an example in this talk, though there's clearly other trisomies we can screen for at the same time. The cause of Down syndrome is an extra copy of chromosome 21. So if the fetus has Down syndrome, it follows that the relative amount of chromosome 21 to all the other chromosomes in the bloodstream will increase. And here's a few sums that talk about the level of increase that we have to detect. So this is a karyotype of an individual with Down syndrome. You can see that down here, instead of 2 copies of chromosome 21, there's 3 copies of chromosome 21. Chromosome 21 is about 50 megs long, and it represents around about 1.3% of the human genome. So if you do the sums and think, well, let's imagine this 10% cell-free DNA sample comes from the fetus. So in a Downs affected pregnancy, instead of getting 1.3% of the DNA in a sample coming from chromosome 21, we'd actually get 1.3 for 90% of it, and 1.3 times 3 over 2 for 10% of it. The 3 over 2 is because we have 3 copies of chromosome 21 instead of 2. And the number comes out is 1.365. So the technical problem narrows down to, if we could tell the difference reliably between 1.3% and 1.36% of chromosome 21 DNA, then we would have a reliable test for Down syndrome. For a while, people worried that there wasn't going to be very much cell-free DNA in the blood, and in the 90s, in fact, there was considerable debate about which was the best way to go, because there was kind of 2 approaches to non-invasive prenatal testing. Some people were following the cell-free DNA route, whereas the alternative technology was to look at circulating fetal cells. But if you look at this little calculation I've put together here, you'll see why cell-free DNA has become so popular. So in a mil of plasma, when we get a mil of plasma, which is the cell-free material from the blood, we find about 10 nanograms of cell-free DNA. And 10 nanograms is about, divided by 3 picograms, is about 3,000 genome equivalents. So in 1 mil of plasma, we have about 3,000 genome equivalents. Now, one of the very convenient features of a cell-free DNA sample is that the DNA is already fragmented. Those of you who understand about next-generation sequencing will understand that you need to normally need to fragment the DNA before you sequence it. But of course, with cell-free DNA, you don't need to do that because it comes ready fragmented. And the average length is about 150 base pairs. Now, the human genome is 3.3 billion base pairs. So if you do the sum, you'll find that in 1 mil of plasma, you get about 66 billion fragments of DNA. So no shortage of DNA fragments in a mil of plasma to work with. And that compares and contrasts the number of circulating fetal cells in 1 mil of blood, which is quite variable. But let's say on average, it's about 6. So with fetal cells, you're dealing with very rare events, but with cell-free DNA, you're actually dealing with very common events. There's a lot of DNA in there. And then as DNA analysis technology improved, and particularly with the advent of next generation sequencing, it means that cell-free DNA has emerged as the preferred technology. So I know that some individuals are still working on circulating fetal cells as a technology, but really all non-invasive prenatal tests nowadays are more or less done using analysis of cell-free DNA. Now, the question is, how many fragments do we have to analyze to detect the small increase of 10,000 sequences? Sorry, the small increase of chromosome 21 sequences. So what this graph here shows, it imagines the situation where we have a 10% fetal fraction, and it looks at 2 distributions. The distribution on the left, in the darker color, that's the distribution you would get if you sequenced 10,000 molecules from cell-free DNA. The mean number would be 1.3%, but there'd be a spread around the 1.3% because of the sample size wasn't so very big. And again, if you only sequence DNA from individuals who were affected with trisomy 21, the mean number would be 1.36%, but there'd be a spread below and above 1.36%. So what we need to do to overcome that problem is to increase the sequence in depth. And what these next slides show is as we go from 10,000 sequences to 100,000 sequences to 500,000 sequences to 1 million sequences to 2 million sequences, as the size of the sample increases, the standard deviation around the mean goes down. And when we get to a couple of million sequences, you can see that there's almost complete separation between the affected and the unaffected distributions. And this is the way that we can tell whether a sample is unaffected or whether it contains a trisomy 21. It's important to note in this graph and the other graphs I've shown you that we do not identify fetal DNA when we find any one DNA sequence, we have no idea whether that one sequence comes from the mother or comes from the fetus. All we can say is that on the average, the numbers of chromosome 21 sequences in the sample are either 1.3% or closer to 1.36%. So it's a statistical technique, and it is not based on the identification of chromosome 21 from the fetus. It's just based on counting the total number of chromosome 21 sequences, comparing them to everything else, and then asking the question, what is the proportion? There's a couple of ways you can do the sums. One is to do a Z-score and just say how different is the distribution, how different is the sample that I've seen from the mean. We do something a little bit different. We calculate a likelihood ratio. So we ask the question, for a given chromosome ratio, what is the probability of seeing this chromosome ratio given that the fetus does have Down syndrome, compared to the probability of this seeing this chromosome ratio given that the fetus does not have Down syndrome. And this gives us a likelihood ratio, which is advantageous because the likelihood ratio can be combined with other prior risks, which are often derived from maternal age, to give a probability of being affected by the condition. Fetal fraction I mentioned earlier is on average 10%, but it moves around, and these next 2 or 3 graphs show the significance of that. So this is the same graph again, 2 million sequences at 10% fetal fraction. But you go up to 20% fetal fraction, it's really very easy to see the distribution changes. So there's a good change between the two distributions. But at lower fetal fractions, the distributions start to overlap, and in that case, you would need to have a higher number of counts to be able to pull the distributions apart. So being able to estimate fetal fraction is very important in terms of generating a reliable result. The other technical aspect of NIPT is that there's essentially 2 different approaches to doing the counting. One is to use a whole genome approach, and the second is to use a targeted amplification approach. Now, in the whole genome approach, we amplify all of the DNA fragments from, we amplify DNA fragments from all of the chromosomes, and let's say we generate about 5 million sequences. And then we count the fragments identified as derived from chromosome 21 and ratio them to all the other fragments. Whereas in the targeted approach, we create a PCR multiplex to amplify specific fragments. Now, the benefit of targeted amplification is you don't need as many sequences because you're just looking at the sequences of the chromosome that you're interested in. And you count the fragments amplified with chromosome 21 specific fragments. So if you think about those 2 methods, they both work, both good methods. The advantages of the whole genome amplification is that the actual library preparation method is low complexity. There's only 4 oligos involved in the sample. And a single reaction allows detection of all the aneuploidies, and it also allows extended screening, so we can add extra tests in the fullness of time. But as you'll appreciate, because we're amplifying a whole lot of DNA that we're not particularly interested in, chromosome 4, chromosome 5, chromosome 6, and so forth, it requires a higher depth of sequencing. So examples of tests that use this whole genome approach is our Iona test, but also the NIPT test and the verified test. The alternative is the targeted approach. What we like about it is it's an efficient use of the sequencing capability, but the problem is that the multiplex reactions to generate the targeted libraries are very complex, with thousands and thousands of individual primers to allow you to get that multiplex to work. And if you want to do additional markers, it requires redesign of the multiplex. And examples of that are Harmony and the Panorama tests. There does seem to be a clinical advantage, though, by the whole genome methods. So these are kind of the published numbers for the failure rate of different tests. Also, in terms of specificity, in terms of sensitivity, in terms of accuracy, the tests targeted versus whole genome broadly similar. If you look at the failure rate, you'll see that the whole genome methods have a much lower failure rate than the Harmony and than the targeted methods. So this doesn't mean that the tests are, the failure doesn't mean that the test is wrong. It just means that there's no result because there's insufficient fetal fraction. So I think on average, we prefer the whole genome methods for 2 reasons. One is this improved failure rate. And the second is, as we think about making the reagents available as an in vitro diagnostic product and doing the QA and the QC on the manufacturing process, it's much easier to do that on a mix of 4 PCR primers than 4,000 or more than 4,000 PCR primers. So real advantages in terms of stability and reliability of the reaction mixes. The other thing is that targeted sequencing is good if sequencing is expensive. And sequencing is expensive, but this is just a blog from last year, and it talks about, something that we all know: DNA sequencing is getting cheaper and getting cheaper and cheaper. So in a way, trying to solve the cost of sequencing problem is not necessarily the right problem to be solving, because it's kind of going to solve itself through the advances of DNA technology companies. So the Iona test then, as I said, is a whole genome amplification method. This graphic here just talks about what it's for. It's to estimate the probability that a fetus is affected with trisomies 21, 18, or 13, and optionally what the fetal sex is. And for the woman and the doctor involved, it really is a very, very simple process. A blood sample is taken, it's sent to the laboratory for analysis, the laboratory performs the analysis, the results are sent back to the doctor, and the results are given to the pregnant woman. So as far as the clinical interface is concerned, it's really a very simple test. However, the technology is much more complicated behind the scenes, and these are the steps of the workflow. First of all, the DNA needs to be extracted from the plasma, and then we need to make a library from each of the samples, and part of the Iona workflow is that this is a fully automated process. It's important that this is automated if you want to process a large number of samples and you don't want an army of technicians turning up every day. And then the completed library is prepared for downstream sequencing. And we use the Ion Chef and the Ion Proton, or the S5 systems from Thermo Fisher. We find those are rapid and efficient and reliable in our hands. And then the data from those data files are picked up by the Iona software, which is in itself an in vitro diagnostic device, which then processes the sample and generates a report for each patient. We have another piece of software called MyniPT, because it's in addition to getting the sample into the lab and getting the result out of the lab, we also need to get the result back to the patient and the clinician, and this portal is a portal for clinicians to allow them to log in, find out that the sample has been received by the lab, find out whereabouts in the lab it is, and then when the report is ready, it is sent back to them, and they can pick it up via a portal. So that's all I wanted to say about how NIPT works. I mean, I haven't gone into every single detail of the library, but I think it's kind of interesting to think about the numbers and the statistics associated with it. Or maybe that's just me that's interested in statistics because I'm a geek or something. Anyway, I hope you found that interesting. We'll talk a little bit now about the clinical performance of the test and also some of the limitations. From a clinical perspective, it's important to understand the limitations, because the tests are very good, they're very, very good, but they are not perfect. So we've done a couple of clinical validation studies with the Iona test, and here's one that was published in UOEG, 437 samples, and in this particular study, the Iona test identified all of the affected samples and none of the unaffected samples, so that was 100% specific and sensitive, though there were a couple of samples where we did not get a result. And likewise, in a retrospective study, again with 241 samples, 100% specific and sensitive of those samples that generated a result. In reality, the Iona test doesn't give 100% specificity and 100% sensitivity. I'm going to explain why that is in a second, and it's nothing to do with the Iona test, and none of the other tests give that specificity and sensitivity. They are not questions of technology, they are questions of biology. But here's a meta-analysis that's published every year by Mark Gill, and in the latest ones, these were the figures for all of the studies involving NIPT. So the old-fashioned test, the way that Downs screening used to be done, had a detection rate of 84% and a false positive rate of 2.2%. Whereas with NIPT, the detection rate, this is for trisomy 21, it's similar for 18 and 13, not exactly the same. The detection rate has gone up to 99.2%, and importantly, the false positive rate has gone down to 0.09%. And I'll show you on this next slide why it's very important of a screening test for a low incidence condition like Downs. It's very rare. Why it's important to have a very low false positive rate. So here's a simulation. Let's imagine 100,000 pregnancies. And I've used for this a prevalence of Downs in 1 in 600. Now in some populations it's higher, in some populations it's lower, but let's just use this one, 167 Downs in the population. And here's what would happen in the combined test. So in this population, there are 99,833 unaffected pregnancies, but with a false positive rate of 2.2%, we get 2,196 pregnancies, which are identified as high risk. Now, these are all false positives, but for each of those false positives, the woman is invited to come back for an invasive test, an amniocentesis, which is at the very least stressful and not completely safe. We shouldn't overestimate the risk of amnios, but they are not completely safe for the fetus, and no one wants to have one unnecessarily. And with the 84% detection rate, we get about 140 cases identified correctly. If you compare that to the Iona test using our false positive rate of 0.09%, you find that the numbers of false positives in this population drops from 2,196 to 90. So that's 2,100 unnecessary amnios, which has been avoided in 100,000 pregnancies. And as you'd expect, also the detection rate goes up from 167 to 160. Goes up from 140 to 165 or thereabouts. So also a good increase in detection rate. So it's easy to see where the clinical benefits are. When we talk about a screening test, we think about specificity and sensitivity. And as I said at the start, the benefit of NIPT, including Iona, is that the false positive rate is very low and the detection rate is very high. But it's not perfect. The reason it's not perfect is there are 5, possibly more, rare biological conditions which can create problems for all NIPT tests. And we at the beginning of the talk, we said that we're not really looking at fetal DNA. We're looking at placental DNA. So if for any reason the placenta didn't match the fetus, we would get the wrong answer. And there's 2 conditions associated with that. One is a condition called confined placental mosaicism, and I'll tell you a little about that in a second. And secondly, vanishing twin syndrome. If it's been a twin pregnancy but one of the twins has demised and it's in the process of being reabsorbed, it can be leaking DNA into the bloodstream and give a false result because you're measuring the vanished twin rather than the healthy or the surviving pregnancy rather. There's also some conditions affecting the mother. If the mother has a partial duplication in a region of 21, 18 or 13, or if she's mosaic for cells for 21, 18 and 13, or if she has cancer, you may get unusual patterns of DNA counts which can confound the software. Probably the most common problem is confined placental mosaicism. It's a discrepancy of cells between the placenta and the fetus. When people have looked using chorionic villus samples, about 2% of samples show some CPM. That doesn't mean 2% of all samples show CPM. That's 2% of samples that were using CVS. So it doesn't mean that 2% of the tests are wrong. But this is a reason why where NIPT may show an aberrant result. So, for instance, on this next slide, we can see 3 conditions here. So here we have an example of where the placenta is positive for a TRIZ. The fetus is normal. That could give a false positive result. And on the right, we have a condition where the placenta is unaffected, but the fetus is normal. and that could give us a false negative result. Now, these are rare events, and they're not, you've already seen what excellent performance figures this technology gives. But the point of making the point I'm trying to make is that when we talk about this not being a diagnostic test, it isn't a diagnostic test. It's a very good screening test, but it's not perfect, and unless, and I'm not sure how it ever would be given these biological conditions. So that's a little bit about the clinical performance. As I said, it's absolutely first class. It's a real advantage, and I really can't see any reason why in the future all prenatal screening would be performed this way. Of course, once we have a DNA sample from the fetus, we're not limited to looking for trisomies 21, 18, and 13. We can look for other things as well. And the question comes up oftentimes, where shall we go next with this technology? Well, first of all, it's worth pointing out that there's quite a lot of differences in NIPT uptake. So one of the changes is that we expect that this will even up a bit. The two territories that account for the majority of NIPT tests, the USA and China. USA are traditional early adopters of new healthcare technology, and with China, I think it was the right technology in the right place at the right time, and it fitted in nicely into their strengths in BGI, in particular strengths in molecular analysis, and some government policies, for example, around the one-child family. In Europe, there's been fairly rapid uptake by the private sector, but as you know, a lot of healthcare in Europe is driven by public policy, and the national health services have tended to lag behind a bit. So the UK will make NIPT available on the NHS next year, but only for patients identified as high risk by the existing methods. France, again, next year, we think at a higher cut-off, 1 in 1,000 for high risk. Holland is currently going to tender. It's gone to tender. They will offer NIPT universally to all women who want it. Germany is lagging behind, a very small study which will be complete in 2019. Spain and Italy tend to be autonomous regions and small individual decisions by regions, and Denmark is available again, only the high risk. So there's quite a patchwork of development of NIPT, adoption by NIPT across Europe. In all cases, the IVD directive, the European In Vitro Diagnostic Directive, which is the overarching agreement, the overarching European directive, which has been translated into national law by all of the countries, including the UK, and we very much hope that we keep this when we do this. I'm not going to say, well, I'm not a huge fan of Brexit. It's fair to say. I'll probably better keep off that. Hopefully, we'll keep the IVD directive when that happens in a year or two. So any test for trisomy 21 is classified as high risk under the IVD directive, and needs to have a CE mark to show that it's been designed, developed, and manufactured appropriately. In the Middle East, we're seeing steady adoption. It's quite a good market, a bit less price sensitive than other territories. In Asia Pacific, we're seeing very rapid adoption. Some territories are very price sensitive, but for Asia, a breadth of menu is important, and I'm coming onto that. So in terms of future directions for the technology, where do we see this going? So I've already said that quite a few national health services are introducing NIPT, but not universally, only for women at high risk. And the arguments for introduction or not in a national screening program are often couched in health economic terms, and the argument goes, the test is better, but the test is more expensive, so we'll provide contingent screening, which offers benefits to some women, but not all. And maybe one of the options, one of the ways to solve this problem is to have a less expensive test. Two directions for that. One is the cost of sequencing comes down, which I think it will. And then the alternative is a kind of a bioinformatic approach, trying to get more information from the same amount of data. And clearly we're working on both of those approaches. I think there's a lot of potential to make NIPT easier, and when I say easier, this is easier from the laboratory perspective, not from the clinician and the patient perspective, and there's significant potential for automation and integration of DNA extraction, library preparation, and DNA sequencing. We often get asked, can you do anything, would there be benefit in speeding up the workflow? Well, the current workflows take 2 to 3 days. If this was reduced to 1 day, it wouldn't make that much impact, to be honest. By the time the sample's shipped to the lab and the doctor's got the report and the doctor's got in touch with the woman, there's extra time added onto that. So I don't see much benefit in reducing the turnaround time to a day, for example. But if you could do testing in 1 hour, that would be significant, as you'd be able to perform the test in an OSCA setting. So OSCA stands for One-Stop Clinic for Assessment of Fetal Anomalies. The lady turns up, she has her blood taken, goes off for the ultrasound scan, and by the time the scan's finished, the DNA data is ready. That would be very good, but I've got no idea how to do that. I think if anyone could come up with a way of doing NIPT in 1 hour, that would be a very good step forward. And then finally, for the last 5 minutes of the presentation, we're going to think about the extension of screening to conditions beyond trisomies 21, 18 and 13. It's certainly possible, but as this is a screening program and not a diagnostic test, it's really important to balance the benefits versus the costs. And I've put a quote in here by Sir Muir Gray, who said he was the director of the UK National Screening Committee. And he wasn't talking about NIPT, he was speaking in general, and he said, all screening programs do harm, some do good as well, and some of these do more harm than good at reasonable cost. And I think that first comment is telling. All screening programs do harm. So we often think that by getting more and more information, we're generating more and more clinical benefits, but we have to be careful because that is not always the case. So an ideal screening test that measures conditions that are clinically relevant, it's specific, it's sensitive, and it's safe. And from a secondary perspective, it's universally applicable, it's quick, it's got low failure rate and fail-safe mechanisms. And I think we can agree that, that non-invasive prenatal testing meets those characteristics. So NIPT for trisomies 21, 18 and 13 is a very good screening test. But we can screen for additional conditions. So we can look for sex chromosome aneuploidies, X0, that's Turner's syndrome, Klinefelter's syndrome, XXX syndrome. We can look for targeted microdeletions, which are associated with certain genetic conditions. Or we could look for untargeted copy number variants. We could look for deletions, or we could look for duplications in the genome in a general way, not saying we're looking for specific changes, but looking for any change. So there's quite a lot of power in the technology to look more deeply into the genome, but it comes with challenges. Some of these are technical challenges in terms of one's to do with sensitivity. As the size of the fragment being looked at, or the size of the change decreases, the numbers of counts, the amount of DNA sequence to identify that change increases. So there's a cost penalty associated with going smaller. Each test that you add in, even though it's for a fairly rare condition, will have its own false positive rate. Eventually, these false positives started to add up. So all the benefits we've done by reducing the false positive rates of 21, 13, and 18 by taking them right down, as we saw in the earlier slide, could be reversed if we keep adding in more and more tests, each with their own small but not zero false positive rate. But I think the biggest challenge is clinical significance. Some of the things that we're looking at, some of the changes, they have variable penetrance. The changes there in the DNA, but it's not exactly clear what it means in terms of the phenotype in real life. And that makes counselling a real challenge. And then sometimes we're going to find novel changes, and they have even more challenges associated with. In March this year, the Nuffield Council on Bioethics came out with a report. You can find it online. It's pretty well written, actually. What they said was that NIPT should be available for significant medical conditions that manifest at birth. It needs to be accurate. But then it said NIPT should not normally be used to test whether a fetus has a less significant medical condition or an adult onset condition, or to find out whether the fetus is a carrier of a gene for any of the medical conditions. And it shouldn't be used to reveal non-medical traits of the fetus, including sex. And the use of NIPT for whole genome sequencing of fetuses should not normally be offered outside of a research environment. Now, I've sort of put that into a different context. I've imagined an instrument called the Magic Gene Viewer. It allows us for more or less free in 20 minutes to look at the whole genome of the sequence. So let's imagine that technology became available. So the technology is the easier part of it, to be honest. It's what we do with it is the challenge. So we'll be able to look at absolutely everything. We can look at trisomies, sex aneuploidies, known microdeletions, all microdeletions, known single gene disorders, or even all genetic variation. And it's a real issue, this, because you have to think what it is you're going to measure. So extending the range of conditions really complicates the counselling. So at the top of this list, I think we have things that are uncontroversial, or at least fairly uncontroversial. A test that says the fetus is affected or unaffected with Down syndrome. Then we might have a test for Turner's syndrome. Now, Turner's has got fairly variable penetrance, so we don't really know what the outcome is going to be, but quite a few people think that's reasonable to offer that. The fetus has chromosome X triploidy. If you have 3 copies of chromosome X, there's probably no phenotype, or there may be a phenotype, but most women who have 3 copies of chromosome X don't know about it. So whether it's a good thing to test for chromosome X triploidy is a debatable point. Let's imagine the fetus has a microdeletion, but it's found out that it maps in a region associated with autism, but the same deletion is carried by the father. What do you do with that piece of information? I really don't know. And then as we go on, I'm speculating down this list. These are not real examples, but they're the sort of examples which could emerge in the future. The newborn is likely to be healthy, but has a 50% risk of developing severe depressive illness by the age of 40. The newborn is likely to be healthy, but has a 25% risk of developing Parkinson's disease by age 65. The newborn is likely to be healthy, but will suffer from male pattern baldness. So it's a difficult time to get this information when you're pregnant, because you need to, if you're given the information, you have to do something about it. And there's only really 2 things you can do, and they both have real problems. One is to terminate the pregnancy, which may be completely unjustified. It may just be on the basis of unclear scientific advice. And the second is to carry on with the pregnancy, but then spend the first year, the first 2 years, the first 5 years, the first 10 years, or even the whole life of the child worrying whether there's something wrong with it from a genetic perspective. So I would certainly agree with the Nuffield report on the need for caution as we take this technology, which is very powerful and has been very useful so far in terms of trisomy screening, and then try to extend it to more and more conditions. So that's the end, really. So over the last 3 years, there's been a major improvement in prenatal screening through the introduction of NIPT. NIPT tests are better than the existing tests, so they're absolutely justified to be used, and they should be used whenever someone wants to know about the status of their fetus. The Iona Test, which I told you about, is a locally developed, globally recognized NIPT, which is accurate, safe, reliable, and available. I think I'm confident to say that over time all prenatal screening will be performed this way, but I think we're also a long way from what the consensus screening panel should encompass. So with that, I shall finish, and then I shall hand back, I think, to Nick or Elizabeth to see where we are with the questions. Thank you.
Thank you very much, Stephen, for that very interesting presentation. We have about 10 minutes left now, so I think we have a chance to handle some of the questions that have been sent in. So I will fire some of these to you in a random order, and we'll cover as many as we can in the remaining time. So the first one was a question that was sent in advance from Anna, and the question is, how do we overcome the limits of detection in circulating free DNA?
Well, I saw that question. I didn't really understand it, because in a way there isn't a limit of detection. There are no, as far as we're aware, I mean, I guess it's hard to prove this, but there's no samples where there's no fetal DNA. So even with a very low fetal fraction, 2%, I think I showed, but in that 1 ml of plasma, there's billions and billions of DNA, fragments of DNA. So the problem is not with the sample, the problem is with the technology. And it's not really a question of any limitation in the technology, it's to do with the cost of the technology. So we can measure more or less whatever we want, but as we go to smaller and smaller changes and lower fetal fractions, we need to have higher and higher depths of sequencing, which will cost more and more to do. So I don't really see that there is a limit of detection issue for fetal, for cell-free fetal DNA in a prenatal context. Now, it's very different when we think about applying the same technology to cancer screening, because clearly if we can detect early changes associated with fetal conditions, it would be reasonable to assume that as a tumour's development somewhere in the body, it will leak DNA into the bloodstream, and we might be able to detect those changes as well as an early cancer detection test. Now, in this case, the cancer fraction could be much less than 2%, 1%, 0.5%, or who knows what percent. So I think for oncology applications, yes, there is a limitation about the limits of detection, and for those ones, I don't know what the answer is.
Okay, very, very good. The next question is one that was sent in through the chat, and it comes from Laura, and her question is, can the quality of DNA extracted from blood samples be an issue to perform the Iona test?
It tends not to be. In my previous embodiment in molecular diagnostics, I worked with oncology samples, and they were terrible. The people, the pathologists would always want to fix them in formalin and put them into paraffin and all manner of insults before you got your hands on them. But one of the things I've enjoyed about working in this field is that the samples tend to be pristine. Once you've taken a blood sample and spun it down, it really is pretty stable. So the quality of the DNA is actually very consistent. The only thing that we have to do in terms of quality is to stabilize the sample. And when we stabilize the sample, there are special blood collection tubes. They are not actually stabilizing the DNA. The DNA is perfectly stable all by itself. What we're stabilizing is the white cells in the sample, and we're trying to stop them from bursting. Because if the cells burst and leak out their DNA, they contribute to the maternal part of the sample, and the fetal fraction effectively goes down. So really, sample quality is not a major concern for us in this particular testing field.
Okay. A question from Federico. Could a qPCR approach focused on the most important aneuploidies reduce the time to deliver a result?
Possibly. It's a real challenge, to be honest. It may be that I'm just living in an old-fashioned qPCR world, but when I used to do qPCR, when we developed qPCR tests, if we could see a twofold difference between two samples, we thought we'd had a very good assay. Even at 100% fetal fraction, the best you can get is a 1 to 1.5-fold difference. So it's a real technical challenge to do this sort of testing by qPCR. If it was possible, yes, it could take the testing time down to say 24 hours. But as I explained earlier in the talk, 24 hours, 48 hours in the clinical context doesn't actually make that much difference. The big difference, you need to be down in the 1 hour, 2 hour ranges, and I don't think qPCR can do that. Or maybe they can, but I wouldn't take it on.
Maybe I try and combine two questions in one, which have just come in because I think they're both related to each other. Anne asks, does the percentage of fetal fraction change over time in pregnancy, and what is the earliest time in pregnancy for non-invasive prenatal testing? And Yanisel asks a similar question, what is the best moment to make the test?
The fetal fraction does tend to increase over time, so there's no upper time limit on having a non-invasive prenatal test. That's a benefit actually compared to the combined test and the quad test and the integrated tests, which were very much set into a specific time block of the gestation period. There's a bit of a challenge bringing the time forward. It may be that we can run forward to 9 weeks or 8 weeks. We don't do that. We say 10 weeks. The reason that we do that is 10 weeks is when a woman knows that she's pregnant and goes off and gets a scan. So 10 weeks tend to be, the earliest you would get a scan. And until you've had a scan, it wouldn't be advisable to have an NIPT test because you might be pregnant with twins. It's probably possible to bring it forward by 1 week or 2 weeks. It may be possible in some cases to bring it forward by more than that. But just, again, trying to put the test back into the clinical picture, it fits in very well with a health system which is almost universal around the world, where women tend to get a 10 to 12 week scan.
Okay, a couple of questions with a more commercial view. Basil asks, what parts of the test need addressing to help lower the cost the most?
It's an interesting question, that, Basil, because 2 years ago, 3 years ago, we would have said sequencing. Absolutely, the big cost is the cost of sequencing. But as the power of sequencing systems increases, the balance changes. And the reason is this, is that on the sequencing platforms we multiplex. We don't just sequence one sample at a time. We use barcode adapters, and we'll sequence 8 or 16 or 32 or 64 or even 96 samples at a time. So the higher the level of multiplexing, the lower the sequencing costs. But of course, during DNA extraction and library preparation, you can't multiplex that. That has to be done on an individual basis. So I think we're in a time now where the balance of cost savings is moving from the end of the process, the sequencing part, towards the front of the process, DNA extraction and library preparation. Though it's fair to say that right now it's moving in that direction, but right now the cost is still how do you get the cost of the sequencing down.
Mm-hmm. And staying on the commercial theme, Linda asks, are you planning, and I assume by you she means Premaitha, are you planning to commercialize the sample prep automation equipment?
Yes, very much so. We already have an automated system, and we're working on a new improved automation system. We think there's room for improvement there, and what we'd like to see is an integrated DNA extraction and library preparation unit that would then feed into the next stages of the sequencing. And I'm sure in the fullness of time, there will be a black box essentially where blood goes in at one end and the result comes out at the other end. And as we look to that, that's not really an advances in sequencing technology question. It's an advances in miniaturization, automation, and system building. But that's no doubt where this will end up.
And maybe time for just one or two more. A question from Richard. Are any point-of-care devices available, or do you see the possibility that this test might move towards the point-of-care?
I'm not aware of any point-of-care devices which are available. The challenge with a point-of-care device is, as I talked about earlier, is the time. It's got to be fast, and there's a tremendous amount of information which needs to be generated in a small time. So, we see nanopore sequences as an interesting technology. Maybe they have the capacity to allow a very rapid test. That isn't the case at the moment. But that's an interesting place to watch out. But right now, it's very much a laboratory-based test.
Okay, and then finally, a question I think was probably answered during your presentation, but another question from Basil. Are whole fetal cells also circulating in a mother's blood? And if so, would that change the performance of NIPT?
Whole fetal cells are circulating in a mother's blood, but first of all, the numbers are really low. And secondly, the first step in the process is centrifugation. So if there were any fetal cells in the mother's blood, they'd be centrifuged down with the other red cells and white cells, so they wouldn't interfere with the performance of the test.
Okay, and I'll just try to squeeze one more quick one in that just arrived. Stephanie asks, what database are you backing your sequence analysis against?
I think it's HG19, but if the question means which human build of the human genome, I think it's HG19, but I couldn't be certain about that. If the other question, there's an alternative question, which is what is the database of the variation of count numbers and fetal fractions. That's a proprietary database that we built as part of the generation of the data to get the IVD certification.
Okay, very good. I think we're out of time. We managed to get through almost all of the questions. As Elizabeth said at the beginning, the ones that we didn't reach we will deal with by email. I'd like to thank everybody for joining this webinar that's sponsored by Tecan. Thank very much Dr. Stephen Little for his presentation. Hope that you enjoyed it and you learned something, and wish you a very nice rest of the day and rest of the evening. Thank you very much.
And thanks for inviting us, Nick. I've enjoyed the chance to speak. Thank you very much.